from SKB, and in this case it's from Birgitta Kalinowski. And her presentation, she's an expert in geochemistry and geomicrobiology, and also the subject of the talk will be on microbiology, the influence on nuclear waste, the so-called MIND project. Please, Birgitta, you have the floor.
Thank you. Can you hear me? I was thinking about Patrick's comment that he had a later version of his presentation, and I'm happy that I did not try to upload a new version because of that. So I'm using a not updated version because I think you will not be able to see much anyway. I didn't see much when I was sitting there, actually. But anyway, I have been given the honor to be a coordinator of the MIND project that actually started as a thought in 2013 in Prague as a working group. And we got together a group of microbiologists and waste management organizations. And we decided to put together an application for the Horizon 2020. And this is from, let's see if I can find the pointer. Which one is that? Yeah, this is from our kickoff in Brussels. This is what we looked like in 2014. I have a much more funny picture when we are looking up in the ceiling because somebody was doing some work upstairs there. But I thought we should be a bit serious, so I took the serious pictures. Okay, the MIND Consortium, we are 15 partners from research, performance assessment and social science. And I think this part with the social science part made the MIND project truly an interdisciplinary project. And it has been really interesting to have that aspect as well. Me being from the natural science part as a chemist myself. 8 countries are represented in the project, and we also have the Implementers Review Board that has been advising us during the project. And there we have caught the interest from overseas, from the US, Canada, and also from Japan. And these organizations have been represented at all our project annual meetings. So we have had that on a yearly basis from... So we have actually had 4 meetings, and the last one was 3 weeks ago, and the project actually ended last Friday. So, well, I guess I'm not the coordinator anymore because the project has ended. The MIND objectives were to try to include microbiology into the safety assessment perspective. It was actually one of our work package leaders that, before he became a work package leader, started this discussion. It was Karsten Pedersen. He discovered that the microbiology was not mentioned at all in the former strategic research agenda. I think they are now currently working on a new one, and I don't know if the microbiology is included in that one. But anyway, we have now increased the understanding of microbial processes in both intermediate level waste repository environments and also in high level repository environments. So I must say that I feel that we have been really successful and lived up to the goals that we had in the grant agreement. Why are microbes so important? Well, they can actually do a lot. They can influence the chemical environment. And they do because they live. They consume nutrients and they excrete exudates. They may induce degradation of metals, cause corrosion, and also degrade buffer, backfill, and cement. They also produce gases, but they also consume gases. So they are microbes, the autotrophs, they may consume a larger amount of gases and the product will be a lesser amount of gases, which can be a good thing in certain cases. They also affect migration of radionuclides. They can cause migration because they excrete chelators, for example, and they can affect pH, so they also increase the solubility of radionuclides. But they also may cause immobilization due to the formation of biofilms where they absorb the radionuclides. So where are the microbes? Well, they are in many places. They are, for example, in the groundwater. The requirement for microbial life, or for all life, is actually water. They need water. As long as there are water and nutrients, they can thrive. We will also introduce microbes in the high-level waste repository with a buffer, because there are quite a few sulfate-reducing bacteria in the clay itself. So the microbes of concern are for intermediate level waste, methanogens, and for high-level waste, sulfate-reducing bacteria. The sulfate-reducing bacteria, they use sulfate when they respire. They use sulfate as an electron acceptor and thereby create sulfide. And sulfide is a corrodant for many metals, for example, copper, as we're using in the Swedish and Finnish concept. And methanogens, they may call, they form methane. But what can you do with the microbes then? Well, you cannot sterilize the whole repositories. What you can do is to make them feel unpleasant, so they are not active. So that is some of the findings of this project that we have presented in our deliverables, but also in some of the papers. The organizational mind project looks like this. We ended up with 4 work packages in the end. I think we originally had like 7, but we have the 2 experimental work packages. The work package 1 is for intermediate level waste, and work package 2 is for high level waste, and the third one is the one that's called integration, communication, and dissemination. So there is where we both include a lot of the results from the 2 experimental work packages in order to communicate within the experts field, and also with the civil society, and also educational, and to sort of present our results to a wider public. Okay, next. Work package 1. The name of that package was Improving the Geological Safety Case Knowledge of the Behavior of Organic Containing Long-Lived Waste. And that one was led by Joe Small from NNL. Work package 2, Improving the Safety Case Knowledge Base about the influence of microbial processes and high-level waste and spent fuel geological disposal. That work package was led by Karsten Pedersen, Microbial Analytics, and the third work package was led by SEKSN, Natalie Leys and Christel Minendogs. Christel is here, so we can, there. So I think you have a poster also, so you can go and look at her poster. And we also have the work package for the project management. The SKB were indeed there, me and Petra Christensen, my right hand. Yes. Whoops. It's quite sensitive, this. Ta-da. Okay. This is taken from the grant agreement where we present the different tasks within the work package 1. The first one was to review the organic waste in all the disposal concepts in Europe. And task 2, organic ILV biodegradation sulfate an effect on the radionuclides. And the third task, in situ microbiology, limitations on growth, heterogeneity, gas generation, and consumption. And the fourth one included modeling. And this is the one that I wanted to change, actually. Well, some of the key findings was that that involved radiolysis and biogradation. It also involved novel radionuclide integrations, gas consumption, methane generation, and also pH limits of specific processes. It has also been suggested for further study to have more of those in situ experiments. I think that has been requested by both the intermediate level waste group and the high level waste group, because having a larger scale with true repository-like conditions tells a lot more than only it acts as a complement to the laboratory studies. So both should be done. So okay. One other thing that came up is the scale of heterogeneity. When you have microbes in a repository, they create micro niches that have another that may have another environment than in other parts of the repository. For example, if you have a cementitious repository where you have generally high pH everywhere, but there might be niches where the pH is lower, and there you may have microbial activity. And also another thing that came up was if you have methanogens, or you try to induce methanogenesis by adding a lot of hydrogen, for example. Methanogens, they require a lot higher hydrogen partial pressure than sulfate-reducing bacteria. So if you have very low amounts of hydrogen and sulfate is present, the sulfate-reducing bacteria will outcompete the methanogens. So if you don't want to have methanogenesis, you should introduce sulfate-reducing bacteria with sulfate. But that may not be beneficial for other reasons, of course. Another thing is also that if you have sulfate-reducing bacteria present, the sulfide itself is actually toxic. Several studies have been done within the project that are relevant to the upper pH limit, for example. It has generally been assumed that methanogenesis will not occur about pH 10, but in the MIND project we noticed that it actually decreases already by pH 8, which is a good thing. What controls methanogenesis? Well, the presence of sulfate, as I mentioned, and also pH and the amount of nutrients, of course. This is a picture that I got from Joe Small, and this in situ experiment in Finland that has been going on for, I think, 20 years now. Yeah, I think it's 20 years. It's a big container with potential waste, or that's simulated waste. It's a lot of tissue material and some cement, not as much cement as we have in the Swedish SFR, for example. So the pH is not as high as it will be expected to be in SFR. But anyway, you can see that the pH decreases very quickly. Those dots there, they are measurements, and those lines, they are modeled ones. And you see that already after, and pH is on this axis, I should say that. That is pH 11, and after already a couple of years, you have a pH that is very close to neutral. And when pH goes down to 8, we notice that we have increased methane generation. Work package 2. has focused on the parts in high-level disposal concepts. I would say that those in the European concepts, they are quite similar, even though, well, the idea is a bit similar, even though the materials vary. They all have some kind of metal canister that is surrounded by a matrix. It could be clay, or in the Belgian concept, cement. And in Sweden, we have both clay and crystalline rock. And in the work package 2, we had the tasks. They are labeled here in this picture. So the task number 1, microbial generation of sulfide in the geosphere. Task 2, microbial induced corrosion of canisters. And task 3, microbial activity in bentonite and buffer. And task 4, microbial degradation of bentonite buffer. And task 5, microbial activity in backfill and plug and seals. And I saw that there are a couple of posters about degradation of plugs and seals and bentonite, if I'm correctly. So we can go and look at them after this session, I guess. I don't know how long the posters will be there, but they will be there until tomorrow. Within the project, we also had the advantage of look at material from the FEBEX experiment, which is a full-scale, or was a full-scale experiment in Grimsel. There was a heater that was surrounded by bentonite blocks, and we had quite a few of samples from there where we had the opportunity to look at the microbial activity. And it turned out that the microbes that had been the closest to the heater, they were not so vivid, so to speak. They had been exhausted by the long time of heating and draft. But in the outer parts of the clay that was in connection with the rock, one could find viable microbes when you put them in the right conditions, and those can be seen here on this picture. So microbes are very interesting in that way that they can be desiccated for decades or even hundreds of years, but then when you put them in the right environment or at the right conditions with a lot of nutrients, they can be revived. And on this picture, I think this is a flow cytometry picture, where you can actually see those. If you just put microbes under a microscope, you can see both living and dead cells. But if you use, for example, this technique, flow cytometry, you can see that the living ones, the thriving ones, they are green, and the dead ones are red. I don't know if that can be seen so clearly, but that's the way to look at them, to see how active they are. Some key findings of the work package 2. The MIND project has identified factors of importance for sulfate production in the geosphere, energy sources, groundwater mixing, and electron acceptors, for example. It's very important when you have gradients that will make the microbes happy, because then they build a constant supply of nutrients and also electron acceptors as they need for breathing. Swelling pressure is an important limiting factor for microbial activity, but not for presence and survival. As I said, they can survive in a compacted clay for many, many years. They are actually conserved that way. But what is important is to see that they are not active. If they are active, they can do damage. So if they are present, that's okay. And I think we need to live with that. We will always have microbes in repositories. Another thing that we found that was kind of interesting is that clay may also act as a sink for sulfide. If you add a lot of sulfide to a slurry with clay, you will end up with not having any sulfide in the solution. It will be all adsorbed in the clay. Okay. And another thing, another conclusion than the usual in situ request is that the microbial interaction with clays is still something that needs to be looked upon, because it's far more complex than we thought from the beginning. Those are basically the same things as I summarized in the previous one, but so I think we could actually continue. The work package 3, the social science part and also the communication and dissemination part, where all the results that have been collected or come out from the 2 work packages have been summarized in the final synthesis report by work package 3. Another thing that the work package 3 has done is they have performed a number of interviews with lay community and also with experts to see how their perception has changed. If you, for example, they have created a movie on the YouTube. So experts have been looking at this together with the civil society and got questions before watching the movie and then after watching the movie to see if things have changed, for example. And those that changed opinion the most was actually the experts, which is kind of remarkable. There have been several lectures and there have been 2 courses that has been organized by the MIND project, a primer course by MICAS and an advanced trainer course by SEKSN. There have been a lot of exchange opportunities between the different organizations and quite a few PhD students have been involved also, so we have transferred a lot of knowledge to the younger generation. We have participated in a number of conferences and also been in charge of organizing a session at the Goldschmidt conference. And we have quite a few scientific papers that have been produced. It's more than 20. And all the work package leaders have summarized quarterly reports that they have sent to me. So I have gotten quite a few of those. We have created 6 newsletters, 2 periodic reports, and we also have a web page that you are welcome to visit, and where you also can download the publications that have come out on the project, and also all of the deliverables can be downloaded from there within shortly. Mind in numbers, well, this one should be updated since it's actually finished by now. So in 48 months, 15 organizations from 8 countries have managed to produce 52 quarterly reports. Actually, 42 deliverables have now been delivered. That was all the deliverers from the project. And 39 milestones have been reached. 2 periodic reports, 6 newsletters, more than 20 publications, 2 advanced courses, about 20 work package leaders meetings have been held, and 4 project annual meetings. And this is from our last meeting in Stockholm, 7th to 9th of May. And we were more than 60 people attending that meeting. And it was a very good meeting, I think. But I will miss you all. Thank you.